So we'll be focusing on metabolic modeling, building a metabolic model, using it, integrating metabolomics and other data into a model. So today is all about modeling, should be fun. And so I thank all the attendees for joining as much as you've been here all week. And today, we're really glad to see you here. I want to do a shout out before the end of the day to all the staff who've been supporting us, Becky and Sarah and Corey, all the comms team. We could not do what we do without you guys keeping us organized. It has been fantastic. All the social media and everything else you guys have been doing, as well as just keeping this, all the Zoom stuff straight, because we couldn't have done it without you. So thank you very much. Scott, you want to add some things?
Yeah, thanks, Leanne, and thank you, Leanne, for everything you've put into the summer school. But I also want to thank the faculty. I've been continually impressed with the engagement, and the presentations have been wonderful. I've learned some things, which is not too hard, but I hope everybody online has learned some things. And I want to remind people also that EMSL is a user facility. So if you want to access these great tools, we have a variety of programs. And I encourage you to go to the EMSL website to look and see how you can access us. But again, thank you to everybody, and I'm looking forward to another great day of presentations. Faculty have just been fantastic, so thank you all. Take it away, Jeremy.
Well, thank you, Scott. So can everyone see my screen, first of all?
Yep.
Okay, excellent. Okay, so today I'm going to be talking about cell factory design. I realize this is the last day, and so I want to make sure that everyone is following along. So if you have any questions, feel free to put questions in the chat window. I'll make sure there's time at each step to answer those questions. And this is actually an interactive tutorial, and so I'm going to go ahead and put a link in the Discord, and the idea is that you can just click on this link and it will automatically pull up a Jupyter notebook for you that you can execute alongside me while I'm executing things. And so I just put that in the Discord. I'll give folks who have access to the Discord a little chance to boot up, and if that works, then I'll put it out also on the Zoom channel so that everyone has a chance to play with it. Okay, but this way we can kind of split up. Not everybody's like booting up all at the same time. That might overload the servers. So I'm just going to give you a quick intro to kind of an outline of what we're going to be talking about today. So I'm going to be describing how to perform cell factory design, but I'm going to use a very, very simple model. And I'll talk about how you steady state analysis of this model, and then I'll focus on some questions that we would like to be able to answer with cell factory design. So how can we use a model to predict optimal theoretical growth rate? And how can we use a model to predict optimal bioproduct yield? And I'll keep going through these questions until to show you that you can answer all the questions that you would want to ask of a real large organism, but we're doing it with a really tiny model just so you can gain some intuitions about that. Okay, okay, great. So Kevin Lynn confirmed that it's loaded. So I'm just going to go ahead and start the presentation. And we got Mariella. So mine's still loading. Okay. I'll give about 2 more minutes, and then we'll kind of launch into the actual presentation. And if I get, I don't know, about 5 people who've managed to load it, then I'll throw the link out into the Zoom window, okay? Looks like Megan made it. Oh, it's loaded for you as well. Excellent. Well, it's still loading here too. Okay. So while we have an hour and a half for this lecture, so. Thank you, Cameron. All right, looks like we have enough people loading here that I'm going to go ahead and put it out now in the Zoom link. So, great. Oops, where's the chat window? There's the chat. There it is. So I'm just going to go ahead and put the that's to the panelists. I think I actually want to do it to the, can I, okay, panelists and attendees. Okay, excellent. Here we go. So now, here we go. So I just put the link out onto the Zoom link. So now everyone else can jump on and see how that works. And seems loaded. Great. Okay. So everything we're going to talk about here today extends to genome scale models. And the only reason that we're discussing this simple model is so that the entire metabolism fits in your head and you can gain intuitions about how genome scale methods actually work. So imagine we have a complete model of all the biochemical reactions in the cell. Okay, so this is, so nutrients A and E enter the cell, and bioproducts D and F leave the cell. And there's a reaction that takes A to C, another one that takes, and then C to D, and another reaction that takes A to B. And then either 2 moles of B recombine to form C and F, or B combines with E to form 2 moles of D, okay? So this is the diagram notation for this model. But we can also show the chemical reactions for this model as well. And it's the exact same thing. So you have reaction 1 produces A, reaction 2 converts A to B at a particular rate, V2. Reaction 3 converts A to C at a reaction rate V3, and so on. Okay, so these R's are the reaction equations. The VJs are the reaction rates or fluxes. And the arrow direction indicates the reaction is irreversible under physiological conditions. These chemical equations can also be represented mathematically as a stoichiometric matrix. And so the way to read this is that over here on the rows, each row corresponds to a metabolite, and each column here corresponds to a reaction. And if a metabolite participates in a reaction, then it will have a negative stoichiometry if it's the reactant of a reaction, and it'll have a positive stoichiometry if it is the product of a reaction. So for example, reaction R2 goes from A to B. So there's a -1 for A and a +1 for B. Whereas over here we have reaction 4 takes B plus E and produces 2D. So does that make sense? We're all along. Okay, cool. So the idea is that so each color, right? So so that is the diagrammatic, the chemical equation representation, and the stoichiometric representation. And now we're going to show you the code representation. So it comes from a software package called Cobra, and you define a model, and then you define all the metabolites, and then you add the metabolites to the model. Then you define all the reactions, and you add the reactions to the model, and then you define the stoichiometries, how the metabolites relate to the reactions. And then I don't know if you guys have ever seen a Jupyter Notebook before, but you can click this run. So as you notice, this cell here is outlined blue right now, that means that if it's blue, it means it's in execute mode. And if it is in, command mode, and this one's in execute mode. So green means that I can actually edit it, right? And blue means that I can execute commands. And so if I click run here, then it's thinking for a while. You can tell it's thinking because there's this black dot right here, and it's thinking real hard. Well, there we go. And now it generates the stoichiometric matrix that we just showed here. So this is now active code, so you can start to manipulate these reactions, and you can add new reactions, you could remove reactions. And the idea is that we want to make the mathematics come alive. Okay, so now that we have a model, the question is, what can you do with it? So like the terrace pools of in the geothermal hot springs of Pamukkale, Turkey, the metabolic network is in steady state. When the metabolic network is in steady state, the concentrations of the internal metabolites do not change. So if I were to write an equation where I say, okay, here is C is the concentrations of the metabolites, and if the concentrations do not change, then we're in steady state. And so the idea is that there's so this dCdt is the change in metabolic concentrations with respect to time, and V represents the reaction rates, also known as fluxes. And S is the stoichiometric matrix that we showed up above. So if you take this matrix here, so this is a kind of the blown up version of that. You have the metabolite concentrations here, and S times V is the stoichiometric matrix, which we'd introduced earlier, times a set of unknown reaction rates. And the claim is that if you're in steady state, it means the metabolites do not change, but the fluxes may. And so the idea is that if you come up to here, you can see that if there's a flux V1, that has to equal V2 plus V3. So you can imagine there's some rate that comes in at say 10 millimoles per hour, and V2 plus V3 together have to equal 10, because otherwise, if this was less than 10, then A would start to accumulate in the cell. And if V2 and V3, their sum was greater than 10, then the amount of A would start to decrease in the cell. Okay. So if you solve these sets of equations here, then you can find out what the V's are that result in this steady state. And so one thing that we can do is we could actually ask, well, how many solutions are there? How many different steady states can there be? And it turns out that there could be a whole lot, in fact, an infinite amount. But it turns out that you can describe this infinite set of steady states with 3 solutions. You can describe any steady state as basically a linear combination of 3 what are called elementary modes. So an elementary mode is a minimal set of reactions that forms a steady state. And it's minimal in the sense that removing any reaction from this set results in a network that cannot achieve steady state. So if I were to remove, for example, V2, then there's no way that if you have flux coming into A, then if you remove V2, then there's nowhere for A to go. And so it's no longer in steady state. Likewise, if I were to remove this reaction, again, I could not achieve steady state. And so I'm just going to go ahead and, so there are 3, like I said, there's 3 different solutions here. So I can go from V1 to V2, and then V5 down to V4 out through V8. Or I can go V1 to V2 and then down through C and F, out, and then up to D and out. Or I can just go V1 to V3 to V7 to V8 and out. And each of those 3 are minimal steady states in the sense that I can't remove any of these reactions. And together, that describes any combination, any superposition of these elementary modes describes a feasible steady state solution. So I'm just go ahead and stop here and just make sure I have, or if there's any questions. So I see there's already a bunch of questions in the chat. Junhoon, did you want to?
Yes.
Yeah. Were there any questions that you haven't already answered, or?
Oh, what was that question? Was about the unit of the fluxes.
Ah, that's a great question. So the units of the fluxes are in millimoles per hour or in millimoles per gram dry weight per hour. So yep.
The next question was, how were these elementary modes detected from eigenanalysis?
So it's similar to eigenanalysis, but there's actually a whole algorithm because it has this additional requirement that that these are minimal, this minimum cardinality, and so so there's an actual algorithm for computing that. In fact, later on in the course, if you stick around for the fluxomics course, I actually have software that calculates these elementary modes for you. Okay, and so it's not actually, I don't think I included that software in this particular notebook, but it is in the fluxomics notebook. And actually, that's a good point. I'll add a note to myself to add the calculation of these elementary modes in a future version of this course. So that's actually a really good point. Any other questions, Junhoon?
Not at this point.
Not at this point. Okay, cool. So, so for the ABC model, all these feasible steady state flux distributions can be decomposed into non-negative combinations of just these three elementary modes, right? So although these elementary nodes are a useful conceptual framework for analyzing small networks, they're actually not practical for genome scale network analysis because the number of elementary modes increases exponentially with the size of the network. So nonetheless, for this ABC model, keeping in mind these three elementary modes will be helpful when solving the cell factory design problems below. So now we're about ready to go into cell factory design. So now that we have a model, and we know that we can create steady state solutions, and we know how to break those solutions down into the component parts, then we want to use this model to predict some interesting questions from the perspective of cell factory design. And the idea here is that, so I'm actually part of a project, as is Junhoon, and actually half the people here in the half the instructors at least are a part of a project called the Agile Biofoundry, and we're really interested in metabolic engineering and trying to trying to basically achieve specific titers, rates, and yields for production of biochemicals, bioproducts, and biofuels. And so these particular questions are of really importance, real importance to us, and it turns out it's important in general for the biomanufacturing industry. And so, one of the critical questions that that converts a toy that's in the lab to something that can be expanded economies of scale is if you can get certain yields out of your bugs. If you can achieve a certain titer rate and yield, then all of a sudden it becomes economically viable, and you can start to displace industries, ways of producing chemicals from petroleum, for example. So, so the questions that we're interested in most are, first of all, predicting the optimal theoretical growth rate. So we want to know how fast can this bug grow given a particular nutrient set of nutrient conditions. The next question we want to ask is, how much bioproduct could it yield in the best case scenario, right? Because if your optimal bioproduct yield is 10 grams per liter, and your funding agency says, well, you don't have success unless you have 20 grams per liter, you can say, well, hey, this is the best it can do, right? The third question is, what is the effect of modulating environmental conditions on bioproduct yield? And so now we're asking, how can we manipulate the environment around the organism in order to improve yield? And then the next question, of course, is, well, okay, if environmental perturbations don't work, can we actually perform genetic perturbations to improve bioproduct yield. And then there's an important question about yield versus growth rate. Turns out that there's an important trade-off that sometimes the faster the more product you produce, the slower the organism grows. And that can be a problem if you are worried about evolutionarily stable solutions where some mutant might and that produces less of your bioproduct might outcompete your strain of interest. So let's start off with the first question. How can we use a model to predict the optimal theoretical growth rate? Suppose the uptake rate of A is limited to 10 millimoles per hour. And let's imagine that D is biomass. Okay, so we can assume the uptake rate is constant if we place the organism in a chemostat where we provide a carbon source at replacement rate. So what is the maximum growth rate achievable given the constraints of this network? So we can find this answer by solving the following optimization problem. We're trying to maximize V8 because V8 is the reaction that takes D and puts it to the outside. So we can imagine coming back here, we're saying that D is like biomass that's leaving the cell. So you can imagine drawing a circle around the cell, and that's the volume. And as it produces more biomass, that biomass leaves that volume and produces another copy of itself. And so we want to know the rate at which this biomass is being generated. So that's the thing that we're trying to maximize here. We're trying to maximize the growth rate, but it's subject to some constraints. And the first constraint is balanced steady state, as we mentioned before. We also have a set of irreversible reactions, and so none of the fluxes can become negative under physiological conditions. And then the last constraint is that the uptake rate is 10 millimoles per hour. And so the most that V1 can generate is 10 millimoles per hour. And so the question is, how much V8 could we, in the optimal case, produce? So it turns out that we can implement this with our COBRA model, and we set the objective here, and then we say that the lower bounds for all the reactions is 0. And then we say the upper bound for R1 is 10. And then we say, what is the optimal growth rate? Well, we'll optimize and see what the optimal growth rate is. And so I'm going to go ahead and click on, so notice that this is green right now. I will just click on it, and it generates the optimal solution. It turns out the optimal solution for R8 is 20. And these other fluxes are required in order to produce it. So I don't know, a list of numbers is maybe not so helpful. So we can also visualize these fluxes. So Escher is a software library that's developed by the folks who basically invented flux balance analysis. And we're going to go ahead and execute that cell. And what you see here is those fluxes overlaid onto the metabolic network. And so you can see that R1 is 10, R2 is 10. But then when B and E combine together, then that produces 2D. And so that's why R8 is 20. Do we have any questions so far? Or does that still feel pretty straightforward? Or we have Junhoon?
We have a few questions.
Sure.
So first one was the Discord link. This is not working. If you go to the link and click the day 5 metabolic modeling discussion, that's where that question is happening.
Yeah.
And another question was about how to calculate the maximum theoretical yield on a certain product using glucose as a carbon source. I think you're gonna touch on that in the next topic.
Yeah, I think I just answered that, but yeah. So if people are asking how to do this with glucose, we can pretend that A is glucose, right? Now, obviously, with a genome scale model, you'll actually have all the metabolic pathways, but the mathematics that describes and the software that actually computes it works exactly the same for this tiny little model as it does for the genome scale model. I don't know if I answered that question. Did Junhoon, did that answer the question?
Yes, and think the second one was for yield. Isn't it computed by dividing flux or product by flux of substrate?
Yes, it is. Very good point. So, so the yield in this case would be 2 millimoles. Well, actually, it would be 2, right? Because you're dividing the units on both sides. So it would actually be so yes, when you do the computation, then it's 2, right? So this is the rate, and the yield would be dividing the output by the input. So it would be a 2-to-1 yield. That's right.
And we have some questions in Discord. How's the concentration used in the model, given that the cell volume changes during the course of a cell cycle?
That's a great question. So how did the concentrations change as a result of the volume, right? So what we do is we assume that we have a constant volume. And so as the organism grows, then that biomass that exceeds that volume basically gets considered like as an output reaction, right? So yes, although your concentrations are doubling, they're happening outside the volume of this particular cell. So the density hasn't changed. And so in this analysis, we assume that all the metabolites are steady state, so they're not changing. But obviously, that is an assumption, or it's a simplifying assumption, I should say. If you want to actually be able to predict how the metabolites change with respect to time, then you can use something called dynamic flux balance analysis, or if you happen to have all the kinetic parameters associated with the organism, which for some organisms that has been done, then you can actually calculate like the Michaelis-Menten dynamics of that cell.
And related question in the chat was, what is the unit of biomass in that case?
Yeah, so the unit of biomass in general will be millimoles per gram dry weight per hour. Yeah.
And then question in Discord, how do you ensure global maximality and not local optima confinement, at least for larger scale systems? Are these stochastic or deterministic models typically?
That's a great question. So, so the question of local versus global optimization. So this is actually predicting things on a bulk scale. So it's kind of scale invariant. So global looks like local, for at least from the perspective of this analysis. And with respect to the question of, can you repeat the second part of the question, Junhoon?
The second part was, are these stochastic or deterministic models typically? In parentheses, I imagine deterministic at small scale, but likely to diverge for larger scale models.
Yeah. So there's actually something called a whole cell model, which has both deterministic and stochastic aspects to it. And so, for example, gene regulation is stochastic, but flux balance analysis is deterministic. But of course, anytime you have stochastic variables, then that induces a probability distribution over your deterministic variables as well. So the whole thing is still somewhat stochastic. So that's a really interesting question. And there is also a particular type of analysis called Bayesian metabolic control analysis that tries to figure out these, which enzymes to increase or decrease in order to improve titers, rates, and yields based on multi-omics data. And that also has stochastic and it has stochastic priors, and then you. So that is I'll mention Bayesian metabolic control analysis and put a link in the chat window. That's a very interesting way of combining deterministic and stochastic modeling together.
Okay, and we have a couple more questions.
Okay, go for it.
How do you make R3 become 0 for this case?
Okay. So in this case, what was calculated was this, as we mentioned beforehand, there are many different solutions, right? So there's this solution, there's this solution, there's this solution. This is a solution that achieves steady state and also happens to be the optimal growth rate. So I didn't make R3 0. We'll be talking about how to do that later on. This is in a perfect world where your bug did was optimized to grow as quickly as possible. What it would do is it would shut down the production of R3 so that all the flux would go to R2, and likewise it would shut down R6 so that all the flux from B would go to D. Okay, but this is obviously not what happens in a real cell. Most cells are not growing optimally. It's a great question.
Another question was, could we add a substrate A supply rate constraint upstream of cellular uptake to this example? For example, the catalytic efficiency of an extracellular enzyme producing A from a non-limiting complex substrate.
That's a great question. So yes, absolutely. So you can imagine adding some kind of extracellular enzyme that produced A from some really large molecule, and then that would probably become the rate limiting factor. So 10 would no longer be the rate limiting factor, but whatever rate that enzyme was able to break down that large molecule. Yeah, that's a great question. A perfect way of extending the model. Okay. So note that, and along those lines, note that R1 is a limiting reagent or bottleneck. It doesn't matter how much E is available, we can only convert E to D at a rate supplied to B. And since they are stoichiometrically constrained by because they are stoichiometrically constrained by R4 to be equal, right? So even if you had unlimited amounts of E, you still would be able to get a higher growth rate because this is your limiting factor. Also, notice that this solution is exactly one of the elementary modes. It's equivalent to this elementary mode right here. Okay. So the next question is, how can we use a model to predict the optimal bioproduct yield? Since we're interested in microbial cell factories, let's think of F as a high value product. So although we called it waste earlier, it was only waste from the perspective of the cell. From our perspective, you know, a microbe's waste is our microbe's trash is our treasure, right? So what happens to the flux distribution and the growth rate if we maximize the production of F? So now we've swapped out V8 here. Now we've replaced it with V9, but everything else is exactly the same. So we're still balancing the steady state. We still have irreversible reactions. And we still have the exact same uptake rate. Okay, so now if we click on this, then we get an optimal objective value of 5. But our objective is not growth rate anymore. It is bioproduct yield. And this is what it looks like. And so now it's going from R1 to R2 down to R6, producing F and also producing D. So it's still growing, but it's now producing your product. Okay. Any questions about that?
Not related to that, but the previous question was about how do you arrive at the definition of flux and biomass unit?
How did we arrive at the definition?
So I said flux is a specific rate, millimole of substrate per dry weight of cell per hour. And
right. That gram dry weight kind of thing. Got it. I understand. So the idea is this: if I were to take the cell and dry it all up and then measure all of the essential compounds that are necessary for growth, you can imagine taking all those essential compounds in their proportions, right? And that would be each of those essential compounds in their proportions would be the left-hand side of a giant reaction. And the right-hand side of that giant reaction would be 1 unit of biomass. And so you can imagine if I were to take all the, that proportion of, I don't know, 10% lipids and, 20% proteins and nucleic acids and, carbohydrates, and I were just to list out all those different chemicals, and I were to do it in the proportion that would be necessary to produce, it's like a recipe for producing another copy of yourself. And so that gram dry weight per hour, if I were then to add up the weight of all those metabolites in their proportions, then that would be the gram dry weight per hour. So that's the gram dry weight. That's where that comes from. And the idea is that value, whatever it is, when you add up all those metabolites in their proportions, equals 1 unit of biomass. And so that unit is 1 gram dry weight per hour.
I'd like to add that the objective that we predict here is becomes growth rate that we can compare to experimental growth rate.
That's right. That's right.
That's all the questions we have so far.
Okay, so good. So now the next question is, how can we model the effect of altering environmental conditions on bioproduct yield? We see from the previous exercise that if the cell wanted to produce the product, then there's a pathway that enables both growth and bioproduction. But cells don't want to produce bioproducts, they just want to grow. So how can we align the cellular objective with our engineering objective? Well, by looking at the difference in flux distributions between optimal biomass growth and optimal bioproduct yield, it seems like there's a decision point at the production of B. To optimize growth, B combines with E to produce 2 moles of D. On the other hand, to produce bioproduct, 2 moles of B split to form C and F. Perhaps we can alter the environmental conditions to induce bioproduction. In the ABC model, let's imagine E plays the role of oxygen in the metabolism of a facultative aerobe. Having some makes the growth rate increase, but is not strictly necessary for growth. So what happens to the flux distribution if the cell tries to grow without E? Would this result in the desired pathway being utilized? So here we have, we've returned back to our cellular objective, that's V8. And we have our balanced growth steady state and our irreversible reactions and our uptake rate. But now we're going to set our E, we're going to set an anaerobic environmental condition to 0. So we'll set V5 to 0, which is the reaction that produces E. So does anyone want to guess in the chat window what's going to happen?
So we have a question. Is that always a growth coupled bioproduct we can model?
No, oftentimes they will not be growth coupled, and you will not be. So you could have 100% bioproduction and 0 growth, or you could have 100% growth and 0 bioproduction. So they won't always be coupled. That's true.
We have an answer.
More bioproduct, right? So that would be the right question. So more bioproduct, someone says, slower growth. Becky Ford says, definitely that's probably true. All right. Anyone else want to hazard a guess? All righty. So let's see what happens. So do we get more bioproduct? And the answer is actually no. So like I said, we asked the cell to grow as quickly as possible, given the fact that there was no E in the media. And so it says, okay, well, give me no E, I'm gonna produce more growth by taking the R3 reaction to the R7 reaction and then out to D. So Becky was correct in that it will grow slower because the optimal growth rate was 20 before, now it's only 10, but it will not produce more bioproduct. It actually found a way to produce growth without having to produce any bioproduct at all. So this is a non-growth coupled solution, right? So the optimal distribution in the absence of E is to convert A to C to D. Okay, so as we saw in the previous exercise, even a simple model can generate counterintuitive results. Perhaps by knocking out the genes that enable this alternate pathway, we can sculpt the metabolic network towards our objectives. So do we need to install Escher separately, or is it within the Cobra package? No, it is a separate package, but if you install Escher, then that will automatically install Cobra because that's a dependency. Okay, so now we still have, we've added one more constraint. So now we're still cellular objective, still have balanced growth rate, irreversible reactions, uptake rate. We have our anaerobic environmental condition, but now we have this additional genetic perturbation. We're saying, all right, sorry, bud, we're not going to allow you to go. We're not going to allow you to take this alternate route here. The only way that you can grow is if you take R2 instead. See what happens there. Anyone want to hazard a guess? Oops. All right. Optimize growth and bioproducts. Let's find out. So the optimal solution with objective value 5. So yes, it's taking this third elementary mode here, right? So you notice that so this is the route that it's taking, and it is actually it is optimizing growth and bioproducts given the constraints of the network. So yes, it's actually just optimizing growth, but in order to grow, it actually has to end up producing bioproduct. And so this is the maximum. This also happens to be the maximum amount of bioproduct that can be produced. Okay, so how about productivity? Productivity is the product of growth rate and yield. By knocking out R3 and restricting ourselves to an anaerobic conditions, we're able to maximize bioproduct yield, but the growth rate's pretty slow. Perhaps we can improve aerobic conditions and see how that affects the trade-off between growth rate and bioproduct yield. Right? So now what we're doing is they're saying we're going to maximize our engineering objective. Nope, actually, that's not right. That should be maximize R8. And that should be the cellular objective. Okay, so the cell is trying to maximize its cellular objective, but now we're going to give the organism a little bit more oxygen and see if that helps it balance out that relationship between growth rate and bioproduct yield. Okay, so just so the only thing that we've done here beforehand is whereas beforehand V5 was 0, now V5 is 5. I'm going to see what happens when that occurs. What do the red and violet arrows indicate? So the red and violet arrows, so here, I'll just show you. So there, actually in this case it's blue. So there's actually a color spectrum going from gray, which means 0, to blue, which is about 5, all the way to red. So it's like a color temperature scale. So the larger the numbers, the more red it is. And also the thickness at the same time is also describing the amount of flux. So it's just more for emphasis. Okay, so back to here. So our optimal solution now is 12.5. So we have, this is growth rate. So we're producing growth at a rate of 12.5, which was more than under the anaerobic conditions. And we're producing less bioproduct than under anaerobic conditions. We were producing 5 before, now we only have 2.5. But if I were to take the product of 12.5 and 2.5, that's going to be greater than the product of 10 and 5. So actually no, 5 and 2.5, right? 5 and 5. 5 and 5 is 25. So back here, it was 5 times 5 is 25. But here, 12.5 times 2.5 is, anyone want to hazard a guess? 30 something, right? Okay, 32.5. Thank you, Andrew. So any other questions about that? Okay, so then moving on. So now we're going to talk about visualizing the trade-off between growth rate and bioproduct yield with the production envelope. So we've discussed the trade-off between growth rate and bioproduct yield at an intuitive level, but we'd like to extend our intuitions to genome scale models. One valuable way to visualize this trade-off is with the production envelope. So let's generate a production envelope for the original unperturbed ABC model. Now, I haven't defined what a production envelope is, but I hope that when I show you how we calculate it and what it looks like, it'll start to make sense. And specifically the notion of an envelope will start to make sense. So I'm just going to go ahead and show you what the production, how we calculate the production envelope, and then I'll show you what it looks like, and then we'll describe. Then I'll explain what it means. So the idea is this: is that we have some iterator that we're kind of moving from 0 to 20, and we're setting the growth rate. So beforehand, growth rate was an objective, right? Now growth rate is a constraint. We're forcing the organism to grow at a specific growth rate, and then what we're trying to maximize is our engineering objective. So we're saying, all right, let's suppose we fix the growth rate at a particular growth rate level. What's the maximum bioproduct that I could produce given that growth rate? And otherwise, all the constraints are the same. So let's just go ahead and compute that. And it's thinking. That's the blue. That's the black dot right there. And here we go. So now we have a chart that shows on the x-axis is the growth rate. That's in millimoles gram dry weight per hour. And on the y-axis is R9, that's our bioproduct. And we see that if we start off growth rate at 0, then you get 0 bioproduct yield. But then as you increase growth rate until you get to a growth rate of 5, that's when you get your maximum bioproduct yield. And then as the growth rate increases, it starts to steal material away from your bioproduct until there's no more bioproduct left at the maximum growth rate. So you can think of this as an envelope that describes the how much bioproduct you can generate in a theoretical sense, given like an optimal sense, given any particular growth rate. So if your organism is growing slowly, then it's not growing. If it's growing as fast as possible, then there's no possibility for producing a bioproduct. But anything less than that, there is at least some possibility for producing bioproduct. Okay, questions about that?
We have a question about the perturbation.
Yep.
Isn't it possible that the fundamental rate constants are also altered under these perturbed conditions? If we knock out an elementary pathway or substrate source, the abundance of enzymes in the alternate pathway might be altered, leading to different rates of conversion. How do we account for that?
That's an interesting question, right? So one of the constraints on the model is that we assume that there's an uptake rate of 0.5, and maybe that's limited, that point, or 10 millimoles per hour, right? And so we assume that's limited because there's only so much enzyme in the cell. And if we were to free up or reclaim some of that protein capacity and redirect it towards our uptake rate, then maybe that would help us achieve overcome some of those limitations. That's a really excellent point. Yeah, and then it looks like does it look like Arvind?
Yes.
Oh, yes.
Questions?
Yeah, go ahead.
Oh, that was Arvind's question.
Oh, that was Arvind's question. Okay. Yes, isn't it possible that fundamental rate constants are also altered under these perturbed conditions? Yes, it is possible. Okay. So how do we account for that? So there is a way of accounting for protein capacity. So there's a number of methodologies that basically adds additional constraints. You can imagine that there's that every flux needs to be supported by, it needs to be supported by some amount of enzyme production. And you can calculate, so if you place bounds on the upper limit of how much enzyme there can be, then that ends up being something that you can start to play with as you remove reactions. Then basically that reclaims some protein capacity. There are also more sophisticated modeling approaches called, there's something called an ME model, which is metabolite and expression model that explicitly captures a transcription and translation. And for that, they also calculate like a dynamic protein capacity and a protein allocation. And if you reclaim that protein allocation, then you may be able to do better under those conditions as well. But Donasian says, so the practical thing to do is to control the growth rate to about 5 millimoles per gram dry weight. Well, it depends. If you are trying to maximize yield, then yes. But if you're trying to maximize productivity, which is the product of yield and growth rate, then you may want to max. So that was this point right here, is that by boosting the growth rate, even though you lose a little bit of bioproduct, the product of 12 of growth rate and bioproduct yield is greater than pushing everything as much to bioproduct as possible. So I was comparing this value here, which is 32.5, versus the product here, which is a growth rate of 5. We have more bioproduct, but when you multiply these two together, you only get 25. So 25 versus 32.5 means that depending on your engineering objective, you may want to allow more growth. Okay.
We have another question from Abby. How does one go from these models to a working microbe that makes your bioproduct? Are the microbes built from scratch, or do you typically engineer pathways that are or into existing bugs?
Oh, no. So at least at this point in metabolic engineering, we are not building bugs from scratch. We are taking existing organisms and domesticating them. Now, there are cell-free methods that basically say, just give me the pathway and the set of enzymes, and I'll produce your bioproduct as much as possible. But that's still very much cutting edge. Any other questions?
The question is, how long does it take to run some of these different types of models at genome scale? For example, standard FBA versus ME versus Bayesian metabolic control analysis.
Great question. So standard FBA is a linear program, so it scales to thousands of reactions and still very, very fast. ME models are also theoretically, they are, well, it depends on how you calculate it. So there's a certain part that there's a pre-optimization that is very expensive computationally, and then the calculation after that is fast but not precise, and so you have to end up going to a quad precision in order to be able to get it, because it's really an ill-posed matrix. There's actually a more recent ME model that is a little bit better posed from EPFL in Switzerland, I believe. As far as Bayesian metabolic control analysis, the challenge there, it's almost genome scale is what the author calls it, because depending on the so the challenge there is in computing the posterior distribution. So I have some priors and I have some data, and so I want to calculate a posterior distribution. And the posterior distribution is actually quite expensive to exactly calculate. In fact, it's intractable. You can simulate it using something called Hamiltonian Monte Carlo, but that is also it takes a very, very long time, but it's guaranteed to produce exact results. You can also choose an approximation method, which is it's called variational inference or automatic differentiation variational inference, and that is much faster, but then you lose some of that accuracy. It's an approximation method. It usually tends to get the means correctly, but it usually underestimates the variance.
We have a related question about the process. How does the choice of solvers affect the results?
Oh, that's a great question. So as long as we're talking about linear programming, open source solvers like GLPK work just fine. When you are doing things that are more about strain design, we'll be talking about that. Actually, I'm pretty sure that Chris Henry is going to be talking about that. This is where you are adding and removing reactions. And so that operation of adding and removing reactions is very expensive. It's a mixed integer linear program. And all of a sudden, it's a combinatorial complexity kind of problem. And that becomes very dependent upon the solvers. And so there are some pretty good open source solvers. Skip is the best open source solver, but if you really want a good solution, you're willing to pay the bucks, or you have an academic license, you can use Gurobi or Cplex, and those are the two best commercial solvers for this, and that's required for when you're trying to do real strain design.
We have one more question.
Yep.
How would we go about maximizing productivity instead of yield, as you just mentioned?
Yeah, so that's actually a challenging question, because it's a challenging problem to multiply two variables together with an optimization problem. And so there are various tricks to get around that, which involves like splitting. You basically fix one, you solve for the other, then you fix the other, solve for the first one, and you kind of keep going back and forth. Junhoon, you probably know some tricks for that. But basically, it's a hard problem. It's a nonlinear optimization problem that there aren't really good solutions. There's heuristics that work in some cases but don't work in other cases. Junhoon, did you want to say more about that?
I think people have tried using dynamic FBA. So you simulate batch growth and see what the end product is in the given time and optimize over that time course.
Yeah.
Another question was the best in the sense of accuracy or something else in terms of the solvers. I think both accuracy and speed.
Yeah, accuracy and speed, that's right. And it's funny because they're all running the same algorithms under the hood, but it's all about the pre-processing. So oftentimes you can reformulate your problem as a smaller problem or a simpler problem. And so you end up solving that problem instead of the problem that you think you're solving. And that actually ends up giving you a better answer. And so, it's like a rat race to try to figure out, who's got the best solver today, versus grow. And it depends on the problem. So, kind of sometimes you have to try multiple solvers before you find one that works best for your problem.
Oh, two more questions just popped up.
Yep.
Can this type of model be easily extended to microbial consortia?
It can, yes. And so, like I said, the linear programming is very fast. It can be expanded to have multiple microbes all kind of interacting with each other and sharing their metabolites with each other. So yes, and then there are the dynamic versions of those, such that if you have constraints on growth rates, then or you have constraints on you have measurements, then you can use that. Or you have particular objectives where you're trying to get them to cooperate or compete with one another, then there's dynamic versions of these. There's also a whole cell models, which you could imagine not only evolving microbial consortia together, but also allowing for horizontal gene transfer and seeing kind of what kind of cooperation and what kinds of so yeah, there's all kinds of interesting simulations that you can do along those lines. Is this also applied to multicellular organisms? It is. So as soon as you go multicellular, then it gets tricky. Certainly there are, certainly higher eukaryotes or lower eukaryotes, you can certainly measure, you can certainly model the compartments. So like there's a yeast model, for example, and there's lots of fungi models. For human, it gets really tricky because, depending on what genes are on or off in a particular tissue makes a big difference. So you have to kind of know tissue specifically which genes are expressed, and based on that, you can create these like submodels of human. And then the question is, how much planning takes place theoretically before testing in a bioreactor? Do you test each model as you go along and adopt the best model? So the model development is actually a really challenging question. It's dependent upon a lot of issues. One of the most important ones turns out to be the database that you use for the metabolite, for your biochemistry database. So later on this afternoon, we're going to be working with genome scale models that have been automatically generated using KBase. And that is something that I can definitely recommend as especially if you're first starting out, you're kind of getting new to flux balance modeling. It's a great way to kind of get up and running with a brand new organism very quickly. And there's a lot more software support that they have provided to enable you to do that. And okay, so Arvind says, as there are 3 elementary pathways and 2 substrate sources, isn't the optimum condition in 5D space? I remember 2D graph of biomass versus growth rate. We're mapping the 5D problem to a 2D problem in that case. I think, so actually that kind of gets at the next question. So I'm gonna come back to that question, Arvind, and I'm just gonna go ahead and move on to the next point, and we'll get back to that.
There was another question.
Yep.
How do we represent plasmid-based constitutive induced expression as opposed to genomic integration of heterologous pathways in engineered strains in the model?
That's a great question. So obviously that makes a big difference, whether it's an engineered strain has been in genomically integrated versus it's a plasmid. For the purpose of flux balance modeling, it's not really, it doesn't distinguish between the two. The way you could potentially distinguish between the two is you could have, because you can have many copies of a plasmid, you could imagine not having having a very high upper bound on a particular reaction that the enzyme maybe in the plasmid is catalyzing, versus in a genomic setting, then it's limited by the expression of that enzyme. So you could imagine that at least at a theoretical level, you could have different upper bounds, but from the perspective of the flux balance modeling, it doesn't really distinguish. Okay. So on to, what are we doing now? We are production envelope for genetic perturbation and environmental conditions. So as we can see, there's an interesting trade-off between growth rate and bioproduct yield for the original unperturbed ABC model. We see that growth rate is required for bioproduct formation, but bioproduct formation is not required for growth. Also note that this production envelope indicates that the optimal bioproduct yield is not evolutionarily stable. So a faster growing mutant can outcompete our bioproduct producing strain. So how does the production envelope for our engineered strain under low aerobic conditions look? So now we're gonna fix V5 to be 5. We're gonna set V3 to be 0, that's our genetic perturbation. And then we're gonna run the production envelope on that strain, and we're gonna see how that strain fares under the production envelope. And notice what's interesting here is that whereas beforehand, the production envelope, basically, as you achieved optimal growth rate, then you could get like 0 bioproduct. But now the optimal growth rate results in a non-zero amount of bioproduct. In fact, it's about 2.5. And so this is the claim, is that this solution, this gene knockout, this mutant here, is evolutionarily stable, because if let's suppose you produced a strain that was producing, I don't know, 2 units of bioproduct, then it would be outcompeted by a strain that produced 2.5 units of bioproduct, because it would grow faster. And so even if you don't get a perfect strain the first time, you can just literally evolve that strain, and something will arise that can grow faster that must necessarily produce more bioproduct. So that is much more desirable from an evolutionary stability perspective. Okay, questions about that, about evolution and or environmentally stable solutions. Oh, it would be great to know what are the other packages that are needed to run this whole tutorial. If you're going to use the binder link, these packages should be available in the kernel. We'll follow up with a list of other packages. Oh, Junhoon answered that. That's great. Junhoon, were there any other questions?
Not at the moment.
Okay, sounds good. All right, so let's talk about flux variability analysis. And I'll actually get to Arvin's question as a result of that. So a number of cell factory design algorithms use flux variability analysis as a pre-processing step. So it's worth taking some time to understand how the problem it's designed to solve, how it works, and what you can do with it. So let's first talk about what flux variability analysis does, and then we'll talk about how it's actually implemented. So although each reaction has explicit upper and lower bounds, sometimes the constraint on one reaction imposes implicit constraints on other reactions. Flux variability analysis is an estimate of these implicit bounds. And why do I say an estimate? Because the implicit bounds may actually be tighter than what flux variability analysis predicts. And this can be seen in this figure, right? So the green shows the actual bounds that are imposed by the flux distribution. And this box here, this parallel pipette, right, forms the tightest rectangle around the actual solution, which is a polygon. So the FVA forms a parallel pipette around the actual solution, which is a polyhedron, 3 dimensions, and in general forms a hyperrectangle around the polytope in n dimensions. The work required to find an exact solution, unfortunately, grows exponentially as in the number of reactions. But for genome scale models, FVA is usually good enough, right? And so I'll just show what happens when you do flux variability analysis. You end up getting upper bounds that are much tighter than the original ones, right? So beforehand, we only had R1 was 10, but in some sense, this one constraint induced additional constraints on all the other metabolites, on all the other reactions, sorry. So what does that look like? There we go. So this is just like the constraints on all of the reactions that were induced by the constraints of the uptake rate. So how does FVA work? Conceptually, FVA works by looping through each reaction in the network and solving for the maximum flux, and then solving for the minimum flux associated with that reaction. So the idea is that we maximize. So we minimize. We go through every single reaction. We minimize it, and then we maximize it, and we see what the differences are. Right. So this is an algorithm which does exactly that. So the question is, what can you do with flux variability analysis? And to answer that question, I will just, whoops. Oh, I didn't define it. That's right. Sorry, go right here, defined FVA, and now we run it. And there it is. So now all of these reactions have constraints on them, whereas beforehand we only had one constraint. And all of these other constraints were induced by this one constraint. But this is useful, for example, if you're trying to model expression data and you want to know, oh, well, how big can this reaction get? And you can use those induced constraints as additional information. So at this point, I realize it's already past 9:30, so maybe we should move on to, or is there a break between lectures now? What's the schedule?
The current schedule is we have a break at 10 o'clock.
Oh, at 10 o'clock. Cool. So we're actually finishing early. Oh, that's right. So it's an hour and a half. And we start at 8:30. Sorry, my math is off. So are there any questions then?
So I can try to follow up with the packages. I think the Cobra, Azure, and Cameo was the only packages that were used in this tutorial. And Plotly.
Yep. So those are the 4. And in fact, I believe if you go to the Agile BioFoundry, there is a there was an environment.yaml. Did I move it? I think I go in the binder here. Yeah, so there's a pip file, and that tells you exactly what you need to install: Plotly, Escher, Pandas, and Rise. I was originally going to do this in a slideshow fashion, so you don't really need Rise. And then there's a post build where you have to do a Jupyter Lab extension in order to make sure that Plotly works correctly.
Okay, we have some questions in typing.
So did I answer your question, Arvind? I guess is the question, right? Oh, so right. So my point was that even though there's all these there appear to be all these degrees of freedom, in fact, the model is much more constrained than the explicit constraints imply, because there's all these kind of hidden constraints or the induced constraints that make the problem much more tractable. That was my response to your original question. Oh, do you have recommendations on methods for fitting reaction rates based on metabolic and proteomic transcription data sets? Yes, actually, there is an algorithm. Well, there's a bunch of algorithms. One of those algorithms is called eflux, and I think there's eflux 2 is I think currently the kind of best of breed in terms of being able to use yes, to fit these reaction rates based on proteomic data sets. Now, the other one is Bayesian metabolic control analysis. And this is not trying to fit reaction rates so much as trying to fit the ratio of two different strains reaction rates. And that's actually really important because, as you guys discovered on days 2, 3, and 4, a lot of these measurements are not absolute. They are relative. And so it's difficult to use relative changes to predict absolute reaction rates. And so you may do better if you have some fastest growing strain and you want to compare the growth or the expression data of that strain against, some slower some slower strains, and then use that information to try to extrapolate to what a faster growing strain than your fastest growing strain is. And so that's that. I would actually recommend Bayesian metabolic control analysis for that kind of problem. But if you're interested in absolute, then you just have to be aware that there's caveats when it comes to making predictions about absolute reaction rates given relative proteomics and relative transcriptomics. Okay, I see there's other people who are typing.
So the question is, we are taking advantage of stable solutions of the network to optimize the process at steady state.
Yes.
There are networks that can be in the chaotic regime. Is there a way to explore these regimes?
Okay, so yes, there are definitely cycles. Circadian rhythm is a cycle, right? And as far as chaotic regimes, I don't know if you want to take that on, Junhoon. One thing that's interesting about metabolism is that it's pretty constrained. You don't get the kinds of nonlinear behaviors that you see, I don't know, with regulation, for example, or signal transduction that you do with in metabolism. But because you can definitely get like chaos and all kinds of crazy stuff with these hairball regulatory networks. With metabolism, it tends to be a little bit more constrained. But if you have a whole cell model, you could actually explore those kinds of questions directly and kind of experiment to see what kind of parameter ranges result in chaotic chaotic oscillations and things like that. That would be kind of interesting. Do you have any thoughts on that, Junhoon?
No, nothing further.
Okay. Yay, Ricardo, that's great. Like that, you are the ideal audience. I really want to take someone who didn't have any experience in metabolic modeling and still felt like you could come away from this lecture having learned something and feeling like you could take that next step. So that's really great. And just to let you guys know, if you do go to the binder, so I'll just go ahead and put the GitHub in the chat window. there are many more. There are many more tutorials. This is just the first of many tutorials. These tutorials were mainly developed by DTU in Denmark, but I highly recommend them as a way to just kind of get yourself up to speed on all the different ways that you can calculate this. Also, later on today, we're going to be doing a lot more metabolic modeling. So if you guys are excited about metabolic models, that's what all of today is about. And then the last thing is, I'm just also going to give a precursor for the lecture I'll be giving at 12:30, which is on fluxomics. So all this is about predicting fluxes, but of course we want to ground this in truth. And so the question is, how do we measure fluxes, and how can we, or given what we can measure, how can we calculate fluxes? And so that will be the next lecture coming up for me. But in between, we're going to have some really great lectures from the folks at KBase, Chris Henry and Jonica, who I see are on the chat, and so there'll be a chance for that. Okay.
We have a couple more questions.
Yeah.
Any recommendation good tools for time course reaction rate data analysis as opposed to simple A versus B groups?
So time series analysis in general. I mean, there's some really good stuff coming out with like neural networks that are able to handle these kinds of time series. But if you're talking more like specifically metabolic modeling, I'm not sure if there's a specific package that I can recommend. Junhoon, can you think of something?
Not really. I've seen some specific tools out there, but in general, Cobra should be able to take time course data, integrate.
Yeah, with metabolic models, yeah, because it's got dynamic flux balance models. Oh, and then Amanda was asking, what's the difference between genome scale models and whole cell models? Okay, so I should be clear. Genome scale, when I say genome scale, I mean genome scale metabolic model. So a whole cell model contains a metabolic model as one component in a much larger system. So it also contains modules for transcription, translation, gene regulation, signal transduction, cell volume, cell division. So all sorts of different aspects of the cell are captured in one model. And so back in 2012, the very first whole cell model was released by Jonathan Carr from Marcus Covert's lab. And then just recently, 2019, an E. coli model was released by Covert's lab. And now they are kind of opening it up to the community, and so I'm part of that effort as well. And we're interested in taking these whole cell models, and so one thing that a genome scale model cannot do that a whole cell model can is the genome scale model is not capable of generating or simulating multi-omics measurements, right? And whereas a whole cell model can. And can do it from like first principles. And so that's going to be really important. That's in the synthetic biology design, build, test, learn cycle, the idea is that you come up with some strain, you put it through an experiment, you generate some measurements, and then you learn from those measurements to try to figure out what you should tweak about that strain in order to get it to grow faster. And if we could virtualize that design, build, test part so that you had a much faster cycle of design, build, test, learn, then we want to use these whole cell models for basically generating digital twins of the organism that we actually care about. And with that digital twin, then we can simulate the multi-omics measurements and say, all right, well, if this is what the multi-omics measurements actually look like, then this is the changes that I would recommend. Did that answer your question? Oh, what's the name / DOI of the 2019 E. coli whole cell model? So it's actually a little hard to find. So that's a good question. Covert.
I can work on that.
Yeah.
If you can answer the other questions in the Discord.
Okay. Yeah. So this is whole cell E. coli release is the GitHub for, but then the paper is actually not linked from there, which is super annoying. So Junhoon will look the paper up. But this is where you can download the whole cell model. Oh, you found it. Okay, cool. So what were the other questions Jin had?
The other question is, and for systems like Bayesian metabolic control analysis, are
Yes.
they amenable to either transcriptomic or proteomic data in
Yes.
addition to the metabolomic data?
Yes, that's so it can take four types of data. It can take transcriptomics, proteomics, metabolomics, and fluxomics. So if you have 13C MFA measurements, it can take that as input, or it can. But you also need these uptake rates that are really important. You also need to know certainly external metabolite concentrations, or at least you can have relative metabolite concentrations as well. That's fine. You can also have relative transcriptomics and relative proteomics. So and it does better when you provide all of them. So it just keeps learning. It keeps doing better the more data that you give it. So from that perspective, it's really quite useful. Okay. Other questions? See, Arvin and Aaron are both typing away. Here we go, Arvin. Can we use this technique to uncover unknown control cycles signaling pathways from omics data? So this technique won't help you with that, although yeah, no, this approach can't really help you with that. There is a paper that I was just thinking of, though, that does a thermodynamic analysis and basically just uses kind of first principles to figure out, what the kind of optimal metabolite concentrations ought to be, and if there's any kind of divergence from that, then that's an indication of regulation. Also, Bayesian metabolic control analysis also has a capability of learning allosteric inhibition, allosteric kinds of effects. And so the idea is that there's a very skinny prior, it's a very tight prior around 0 for any metabolite to affect a reaction that it's not part of. But it allows that possibility. And so if the data suggests that there should be regulation going on, then it will find it and it will assign a posterior, a non-zero posterior distribution on the effect of some metabolites on a reaction. And so yes, you can use this kind of multi-omics measurements to discover hidden controls. And so there's a few caveats with that. Sometimes in a linear pathway, it can't distinguish control on an upstream versus control on a downstream, but there's actually ways of dealing with that as well, using mediation analysis from causal inference. But that's going down a very rabbit hole. If you're interested in that, though, definitely follow up with me. I can point you to that. Let me point you to the paper, at least, with Bayesian metabolic control analysis, if someone hasn't already put that link in the chat window. Here it is, Bayesian inference of metabolic kinetics from genome scale data. It is. Cool. Jonica, did you want to give a little blurb for your upcoming talks?
I'm sorry, Jeremy, I missed it. What was that?
Oh, I was just going to say, did you want to give a quick little blurb for what folks can expect after the break in terms of what you'll be talking about?
So I'll be talking very broad, a broad overview of metabolic modeling in KBase.
Okay.
And also time permitting, will show a live demo of the workflow that's what the students get exposed to this afternoon.
Great.
And so if anybody interested to see these workflows or try out Keybase, we have sent an email yesterday to sign up for an Keybase account, and we would appreciate if you can sign up for an account so you'll be ready by the time of the presentation.
Awesome. And what's Chris going to be talking about?
Chris is going to be talking about more in depth of the omics data integration, metabolomics integration
Awesome.
into models.
Oh, that's really exciting. Okay, great. So can you share a couple of papers where this type of model has been successfully implemented and validated experimentally if possible? Yes. My goodness, where do we even start? Probably just go to the publications list of Bernard Paulson would be a good place to start. The gcrg.ucsd.edu, does that still work?
I'll try to put some review papers
Yeah,
in
put
the
some
chat.
review papers in there. That'd be great. That's a couple of good examples would be good. Okay, fair enough. So are you interested in metabolic engineering, or like metabolic engineering kinds of papers, or are you interested in more like what can metabolic models do for you kinds of questions? Because there's that review paper, which was basically like it looked like a phylogenetic chart. It was like a of all the different methods that one can apply to a metabolic model. You know which one I'm talking about?
Yes.
Junhoon? Yeah.
I'll put that in the window.
Yeah. That's a really nice kind of overview of all the different kinds of things that one can do with the metabolic model. Oh, but both. I was also hoping for something that gives a sense of the experiment model workflow. Okay. Yeah. So how do you work with? So this is actually something Junhoon and I are actually doing on a daily basis. Is working with experimentalists to try to figure out what strains to generate based on the multi-omics measurements that they've been making. And yeah, so that's the so Junhoon just put the link in there to metabolic genotype phenotype relationship. Did you want to put the one in there about OMG, just like, because showing like how you can do the, you know which paper I'm talking about, the Hector Garcia Martin one, OMG Hector Garcia Martin. Let's see if I can find it here.
Multi-omics paper.
Yeah, the multi-omics paper. That's a good one as well, I think, because it really describes the workflow and emphasizes that. Go ahead and put that one in there. And then there's also one by Paulson. Elizabeth Brunk, I think, was the first author. Brunk. Elizabeth Paulson. Let's see what that one is. Characterizing strain variation engineered E. coli using a multi-omics-based workflow. I think this is the one. Yeah, that's right. Yep, this is it. Excellent. Many thanks. Yay. All right. Well,
I'm impressed with how fast you can find papers that you remember. Like, I remember papers and it takes me like, I don't know how long to find them. So that alone is impressive, Jeremy.
Thanks, Leanne. So we're getting towards the top of the hour. I definitely want to give people a chance to take a break and step away from their computers for a little while. So unless there's any kind of burning questions, I say let's take a break.
Yes, thank you so much, Jeremy, Junhoon. You guys were great keeping track of all the questions. The engagement was wonderful to see. If there's more questions, I'm sure folks will be happy to answer them. But in the meantime, thanks, everybody. We'll be back in a half hour with going through Keybase.
Yay.